Hello everyone, I'd like to introduce you to my poster correcting 4SU induced quantification bias in nucleotide conversion RNA-seq data. In recent years, RNA-seq techniques have made major advances, including the use of metabolic RNA labeling to introduce time-resolved component to RNA-seq data. One of these techniques is called SLAM-seq and uses 4-thiouridine, or 4SU for short, as a uridine analog which is incorporated into newly made RNA. After labeling, 4SU is further chemically modified, which leads to it being sequenced as a cytosine rather than uridine or thymine. And the newly synthesized RNA can then be distinguished by their higher than average T2C mismatches. It has been shown that 4SU can have cytotoxic effects during very long labeling times and high concentrations, but its effect on expression estimates has not been studied, and so we wanted to find out if it has an effect, and if so, how to quantify and correct for it. And this is how our software GrandRescue came to be. So theoretically, 4SU could induce quantification bias metabolically by inhibiting RNA polymerase or by destabilizing RNA transcripts and leading to a quicker degradation. On a technical side, it could inhibit reverse transcriptase activity during library preparation, and on a computational side, by exacerbating read mapping due to an increase in T2C mismatches. So we check this by creating several data sets of increasing labeling times and concentrations in various cell types. And I'll show you more of them later on, but the general trend we could see with higher concentrations or higher labeling times is this. So in this dropout plot here, you can see all the genes ranked by their RNA half-life on the x-axis, and on the y-axis you see the log fold changes in between a 4SU sample and no other perturbation and a no 4SU sample. And you can see that towards lower half-life genes there seems to be a downregulation. We also created a SLAM-seq simulation where we took read sequences and introduced T2C mismatches into newly made RNA according to fixed rates from 5 to 25%. On the left you can see the estimated incorporation frequency by GrandSlam, a software package previously developed in our lab. When we introduce the mismatches into already mapped reads and then estimate the incorporation frequency, which you can see here for the solid points, you can see that everything is fine and the estimation fits. But if we introduce mismatches and then map the reads again, we can see major underestimation of the incorporation frequencies that is just due to the mapping. On the right, we can also see this effect in a dropout plot. Here we don't compare 4SU to no 4SU samples, but instead we compare the 25% simulated rates data compared to the ground truth of 0%, so no introduced mismatches. And you can see that especially for the lower half-lives, you can see major factors like MYC or Jun or EGR and others being downregulated. To remedy this, we decided not to create a new mapping tool for nucleotide conversion-seq data, but instead adapt the mapping strategy. You can use any mapping tool you like. For our lab, this would be STAR, and map the reads as usual, as you can see here. Then you'll end up with some unmapped reads that are then mapped again with the same tool to what we call a 3-letter pseudotranscriptome. We basically take the reference genome, remove all intronic regions, and do a full T2C conversion, so these mismatches do not disturb read mapping anymore. The newly mapped reads here are then mapped or calculated backwards to the original reference genome, and the full dataset can then be analyzed. We could show that our method surpasses other read mappers like STAR, HiZ3N, and SlamDunk in the number of unmapped reads, in the number of uniquely mapped reads, and also the mapping accuracy of those reads. And additionally, comparing the incorporation frequency plot here before and after rescue, we can see that this rescue procedure completely remedies the original problem that we had, and also for the dropout plots we can see that for the rescued part there is no underestimation of genes anymore. Now, comparing our simulated data and biological data, we could see that the effects in real data sets are more drastic, letting us to believe that next to the mappability problem, 4SU also induces quantification bias on a metabolic or technical side. And since this effect is always well correlated with the RNA half-lives, we decided to upscale underrepresented new RNA reads by the correlation factor. And we can see this here as an example. This is what it should be after the correction process. As I've said before, we created a labeling dataset with increasing 4SU concentrations in 3 cell lines and could quantify the bias and also correct for it with this upscaling approach in all the cell lines and concentrations, as you can see as an example here for the 800 micromolar example, but also for all cell lines and all concentrations down here. The same is true for our progressive labeling dataset with 2 replicates. We could quantify the bias and completely correct for it. And if we're now looking at specific genes, for example DUSP4, this seems to be downregulated in total RNA over those 2 hours, but after correction the effect is gone. And the same is actually true for EIF3L, where there seems to be an upregulation, which is due to normalization of the biased counts. And this effect is also gone after the correction. And now if we're looking at volcano plots of all the genes, we could see that before correction a lot of them are significantly downregulated and they are mostly gone after correction. And then finally we plotted the log fold changes from replicate A against replicate B, and we can see this strong correlation here, which is completely gone after correction. And this leads us to believe that the only significant contributing factor for this quantification bias is the dependency on RNA half-lives, which is countered by our upscaling method. So, to summarize, we could show that 4SU doesn't use a quantification bias with higher concentration and labeling times, and this effect is both due to exacerbated read mapping and what we think is an inhibition of the reverse transcriptase activity. I couldn't show you the info here for this theory because of time constraints, but you can find it here in the preprint and check it out there. And finally, with Grand Rescue, we can completely remove the mapping-based and technical quantification bias, enabling a bias-free analysis of nucleotide conversion RNA sequencing data.